sorry, call, there was a call. So what I did was, see, I was actually, this is the main process, okay. um, if I if the RNA gets out from the nucleus without the post transcriptional modification, na, then the RNA will, see here, if the RNA is breakdown, so there will be no translation, the RNA will break. But if the post transcriptional modification done within the nucleus itself, and then the RNA is getting out, see, the capping is also done, tailing is also done, then it will be translation. This is the and this is not the mRNA, this is the heterogeneous RNA, because exons are present, introns are present. See, I have written here, see, due to the presence of introns and exon, it is called a heterogeneous nuclear RNA, this is the hrRNA. Okay, after post transcriptional modification, which is capping, tailing and splicing, means removal of the introns, it is called the mRNA. Now this mRNA will be transcribed outside the nucleus in the cytoplasm. But if the hnRNA is without post transcriptional modification gets out in the cytoplasm, it will break down and there will be no translation. So transcription inside nucleus and translation in cytoplasm. So this remembers this point. Now here we can see that in this one, in case of prokaryotes, post transcriptional modification is absent. There is no post transcriptional modification, because transcription and translation occurs in the same place, so they don't need any protective or exporting the structure. The capping is done to break down, to get out from the nucleus, so that doesn't get down degrade. See, I have told initial in here na, see here it is getting degraded because there is no post transcription modification. But as soon as the post transcription modification is done and then it is getting out, there is no breakage, there is no degradation. So in case of prokaryote, as it is occurring at the same place, so coupled transcription and translation, they don't need any kind of post transcriptional modification. But in case of eukaryotes, the post transcription modification is needed, because the transcription is occurring inside the nucleus, is located inside the nucleus, okay. But translation is occurring in the cytoplasm. So see here, in the different location, it is written occur in the different location, so that in a protective from the cytoplasmic cytosolic enzyme, there is an enzyme that will degrade the hnRNA. So capping tailing is needed, post transcription modification is needed in the case of eukaryote. Okay, see, they need structure for the transport also, they have introns. So yeah, these are all this is the capping, tailing. For a transport you need tailing, for the protection from the cytoplasmic enzyme, for the cytoplasm protection, for protection we need capping. For transportation, outside of to be taken, to be get out from the nucleus into cytoplasm, we need tailing. And to you also need to remove the introns, so that the exons can come together and form the mRNA, which is getting translated into the protein.